Hi everybody, this is Mr. Rittner, and in this video lesson I'm going to be trying to walk you through how to do the actual data analysis with our primary productivity lab, right? Specifically, I've given you this spreadsheet for student NPP data. You'll see that it has a few different tabs at the bottom here. This first one goes over some instructions, so it's trying to walk you through, every time you see yellow here, it's trying to highlight some important instructions for you. That way, while you're working, it's kind of just designed to be a quick reminder about what you're supposed to be doing. But the general idea here is that, you have different formulas and stuff that we can apply. You could do the data calculations by hand, but I think it's to your advantage to learn how to navigate in Google Sheets or even better, Microsoft Excel. But that instructions page is just to kind of give you some background on what we're doing. You know that we're going to be calculating things like the average for our data sets, the mean, if you will. And then from there, we'll also calculate something called standard deviation. Standard deviation is a statistical analysis where we're trying to measure the variance of our data sets. So if you think back to the beginning of the school year, maybe Module 3, remember we talked about accuracy and precision. And so standard deviation allows us to sort of quantify precision, the reproducibility of our results. If we ran this experiment multiple times, do we get the same value every time? Standard deviation kind of gives us an explanation of that. We're going to use 2 units of standard deviation, hence the formula you kind of see here. I'll walk you through that later, but that's because we want to have 95% confidence. Without getting into all the explanation for that, we need 2 units of standard deviation. Once we have standard deviation, we can actually go ahead and calculate standard error, or what I abbreviate as SE. Standard error, you'll see the formulas right there. We'll talk about that later on, but really that's used for us to calculate error, to develop our error bars, and then we can analyze our error bars to see the significance of our results. And you have some extra support here with error bar analysis, but bottom line, if error bars are overlapping between different data sets, if they're overlapping, that's saying that there's no significant difference. It's probably just due to random chance, if you will, that the values appear to be different, just because of the variance within the data set. However, if we're seeing data values and the error bars do not overlap, that means that random chance can't really explain that. Variance is not enough to explain the difference between the data sets, and therefore the results are significant. That's my quick 30-second overview, if you will. So with that said, let's navigate over to light availability. This is going to be our demo one, right? I'm going to do light availability with you. You're going to have to pick for homework either the water availability or CO2 availability data set. You're kind of just doing the exact same thing, but it'll be your responsibility to go through that and submit your table, your graph, and your captions along with that. Okay, but just to kind of walk you through what does this look like. I very intentionally set up this spreadsheet the way that I did so we can go ahead and calculate everything we need relatively easily. So you'll see at the top here in blue, we have plants that are grown in light. This is our light availability treatment, if you will, but we have plants that are grown in light because we need to be able to calculate things like NPP, and then another set of plants grown in darkness because we wanted to calculate respiration, just like we talked about in class. You'll see here for the light plants, we have some data here. I have R1, 2, 3, 4, and 5. So R is just for replicate. We wanted to have some multiple samples within our data set. So I was able to give you the initial mass of each replicate under the 25 lumen condition. Lumens are just a unit of light, so we can vary our light treatment, if you will. But this would sort of be like the lowest level light. 50 lumens, 75 lumens, and 100 lumens. So these are our initial masses, and then right below we have the final masses. So remember, we can calculate the change in mass, and that will tell us NPP effectively, how much did that change over the course of that week. And we'll be able to do the same thing with the dark plants later on. You have the initial mass for each replicate and the final mass right here. Now you'll see that I kind of pre-programmed for you a little NPP table here. And it's designed so that you're going to have a place where you can enter for the 25 lumens condition for replicate 1, what was the change in mass of that particular plant. We'll have all 5 replicates in the same column so we can calculate the change in mass, the standard deviation of that data set, and then finally standard error. But we're getting a little ahead of ourselves. Why don't we go ahead and take a look at the 25 lumens sample. Now, I could sit here and calculate by hand. I could say that, 4.7 minus 2.7, that's 2, it works out to 2, so that would have been a change of mass of 2 grams. That's relatively easy math, but that's going to get really tiresome really quickly. A faster way to go about this is to program a formula. So the best thing I can do is select the cell that I'm interested in. In this case, it's going to be cell C27. And for R1, under the 25 lumens condition, I'll program the following formula. Anytime we're starting a formula, we have to use the equal sign. And that basically tells the computer, hey, pay attention, I'm going to program a formula now. What I could do is I could program those numbers again. I could just type in 4.7 and 2.7, but actually what I would much rather do is program the cells. And that way that'll allow me to replicate the formula throughout, and it'll save us a lot of time. So what I'm going to do is I will select, I've typed in equals, there's my formula being developed. But I want to select this cell right here for R1 under the 25 lumens. So equals B10 is the coordinates, B10. And then I want to subtract, so I'll use the minus sign. And then I'll select the other cell, which was the initial mass for 25 lumens for replicate 1, which works out to just be B3. So ultimately, my formula is going to go into this cell right here. I think that's C27. Should read as equals B10 minus B3, and then I'll hit enter. And look at that. It gave me a response of 2. It actually calculated that perfectly. Now, I could continue programming that same formula over and over and over again, or I could autofill it. You kind of saw that before I clicked out of it. But the best way to do this on the computer is, again, select the cell that you're interested in. It has the formula. We see it's up top there. If I mouse over to the bottom right-hand corner, I get the crosshairs. If I left-click, hold, and drag down, it'll apply the same formula all the way down the rest of the column. Of course, the computer is pretty intelligent, and it knows it doesn't want to do exactly B10 minus B3. It's intuitive, and it knows to go to B11 and B4. Since I'm dealing with R2, that means I'm going here to replicate 2 for the final mass of 25 lumens, so 5.5, and then it's subtracting from cell B4, which was replicate 2, but that was the initial mass under 25 lumens. So that works out really, really nicely. Even better, if I select all of the cells that have the formula so far, I can actually autofill again all the way across. And now, just like that, I have all 20 cells done. I kind of walked you through it, so it took a minute, but once you get the hang of it, you can do this in just a matter of seconds. So just like that, we actually have all the change in mass values for all the different replicates for all the different light conditions. So we're doing great. Now, you'll see that the next part of this table calls for us to calculate the mean change in mass. We want to know the average, in other words, right? Mean means average in this context. The way to do this is pretty simple. We're again going to program a formula. So I'll type in the equal sign here. But rather than actually manually typing in the formula, I'm going to actually use one that's pre-programmed. So if I type in AV right away, it starts popping up with some guesses as to what I want, and I want the average formula. So I'll go ahead and left click on that. So you'll see it says equals average and then open parentheses. That's telling the computer that you're going to do an average for the following cells. You can type them in manually, or you could select them with your mouse like I'm doing, but we want to get cells C27, like you see there, all the way through C31. So if you wanted to type it in, it would be capital C27 colon capital C31. Make sure to close the parenthesis, and then you're done. You've got 1.4, which is exactly the average we were looking for. Again, I can autofill that across the row, and good. We're done with calculating the mean change in mass at this point. Perfect. Now we're going to go ahead and calculate standard deviation. Now you'll notice there is a yellow formula bar here, or a cell if you will, and it's telling us to use this formula. We're going to do 2 times standard deviation dot s, right? That's a pre-programmed formula, but we want to use 2 times that value because I was saying before we want 2 units of standard deviation. So just trust me on this. We want to do that formula exactly. So what does that look like? Well, in cell C33, we'll again click on equals. We're going to go ahead and type in 2 times, and then we want to pull up that standard deviation pre-programmed formula There it is, standard deviation dot S. That dot S is referring to the fact that we have a sample. So we'll go ahead and select that. We don't have the full population. We only have 5 replicates. So that's the most appropriate formula to use. Again, you'll see that there's an open parentheses, so we have to tell it which cells to work with. And we'll just do the exact same ones. Our samples are going to be from C27 through C31. Close the parentheses, hit enter, and our standard deviation value should be returning as 1. Fantastic. Once again, we can autofill across the row like I just did, and we're almost done. Our last thing to do is calculate standard error. You'll again see that the formula is right there, so if you're wondering where I got that from, there it is. So again, we'll program with equals. I don't want to type in standard deviation. Instead, I'm just going to grab the value that was already pre-calculated. So I can go equals C33 divided by, and I want the formula for the square root, and then I need count, so I'll type that in. And then again, select my sample, C27 through C31, close parentheses, equals, and it's 0.4, which is exactly what we wanted it to be. Autofill across, and we're done with the light plants for the moment. We'll do the exact same thing for respiration, the dark plants, if you will. So once again, program our formula. That's going to be equals final minus initial for replicate 1. And I will autofill this all the way down. Notice that this time, all of our values come back as negative. That should make sense. When the plant is grown in darkness, photosynthesis is off, so there's no primary production at all. However, respiration continues, resulting in a loss of biomass. Now, we want to calculate our average again. Just take note that we need to make it an absolute value, since respiration is technically always a positive value, so we will make that adjustment. What I will do for that is I'll program it as equals open minus 1 times, and now I want to do the average formula, so I'll pull that up. We're going to do the average from C37 through C41. That's our sample there. And if we've done this right, it'll work out to 1.7, which is, again, perfect. That's what we wanted. It would have come out as minus 0.1. We just had to adjust it to the absolute value, so I multiplied by minus 1 to adjust for that. Standard deviation should be the same thing as we were doing before, 2 times standard deviation dot S. Select your sample again, close, 1.8. And finally, we're just going to do standard error. So we select our standard deviation value. Don't program that formula again. There's no need. Divided by square root. Count. Select our values. Close. Enter. 0.8 is the right answer. So if you've been able to follow along, that's how you would calculate the data when you go to do water availability and CO2 availability. You'll see at the bottom, I already auto-filled it in for you. But I just migrated all of the data into the corresponding fields to make a nice summary table. So for each light condition, we have our mean NPP value in grams per 75 centimeters squared per week, the standard deviation, and the standard error. We can do the same thing for res, as well as GPP. All I did, though, was I'm just copying and pasting those values. However, it's important to make sure that when you do copy, don't do paste. You want to do right-click and then paste special, paste values only. Because watch what happens if I try, like I'm just going to do an example. If I wanted to take this number, actually no, let's do this one. The mean change in mass for our res. So this was going to go right here. You can see it's supposed to say 1.7, but if I just do paste, it comes up with some crazy thing because it moved the formula with it, and now it's calculating a brand new number. So let's undo that. Instead of just doing paste, let's do paste special, value only, and it comes back as 1.7, which is what we were looking for. Just keep that in mind when you go to do your table. I did it for you on the demo. Now at the bottom here, you'll see an example. This is what the graph should look like by the time you're done with it. How did I get that? Pretty straightforward, actually. All we're doing is we're plotting mean NPP. You're not doing the entire table. I just want to see mean NPP for for the 4 different conditions. I did add in error bars, and you can see that since none of them overlapped, all of these results are significant. In other words, 25 lumens of light returns a different NPP compared to 50, which is different from 75, and 100 lumens. So all of these values are significantly different, which says that as light increases, so does mean NPP, which makes sense. Light is a reactant, if you will. It's not really a reactant, but it's a necessary ingredient for photosynthesis and primary productivity. So anyway, how did I come up with this graph? Pretty straightforward. All I'm going to do, I'm going to highlight right here, because again, I'm just plotting the mean NPP values against the light availability. So I'm just highlighting those 2 rows right there. It's really just 10 cells. From here, I go to Insert, go Chart, all right? It's automatically given me this, which looks similar, but a little bit different. This is not going to be our final graph, essentially. First things first, I don't know why Sheets does this. It's one of the downsides. There's no way to get rid of this value, which is unfortunately just labeled in the wrong place. So what I'll do is I double click on it, and in my chart editor, I can switch the text color from auto to white. So it's kind of, we're pretending it's gone at that point. I do need to label both axes, so why don't we do that? In Chart Editor, I can go to Chart and Axes Titles. I don't want a chart title, that's going to go in your caption, but instead I want Horizontal Axis Title, and this should just be Light Availability, right? Because that explains why they're different colors, right? These different colors mean that each one is its own unique series, because we have 4 different treatments essentially going on. Make sure you do have the 4 different colors. There should be 4 different series. That's why I set it up the way I did. Otherwise, you will not be able to add your error bars in a couple of moments. Before we get there, though, let's make sure we do our vertical axis title. I'm actually going to be kind of lazy, and I'm going to go back here. I'm going to grab my units. Actually, I'll just grab the whole thing. So I can copy that, come back here, customize, vertical. Let's go ahead and just paste that in there to save myself a little bit of time. It should be mean NPP with the units grams per 75 centimeters squared per week, and we're good to go. Let's see if there we go. Little technical piece. So believe it or not, we're almost identical to what the final product should be. What we need to do now, I'm going to give myself a little bit more room, is I'm going to use these standard error values in order to go ahead and program the error bars. All I'm going to do is double click on the series I'm interested in. So I double clicked on the blue one for 25 lumens. That brings me to series. I'm already at 25 lumens. We'll select error bars right here. It does put them in error bars automatically, but it did it as 10%, and that's not what I want. I want to select, instead of percent, I want to put in the value. So I want a constant value, and the value I'm looking to enter would be 0.4, because that's the standard error for 25 lumens. So, excuse me, my value will be 0.4, and there we go. Right away, there's my error bar. Nice. I'll do the same thing. I'll go back up, do 50 lumens. That value should be 0.3, so I want error bars, constant, 0.3. Same thing for yellow or orange, whatever, 75 lumens, error bars, constant, and then 0.6. And then finally, we'll do 100 lumens, error bars, constant, and that should be 0.5. And just like that, we've now programmed our error bars, and you can see that the one we just made together is identical to the one I left you in the example. So that's it. That's how you go ahead and program these sorts of things. When you're done, you'll just sort of copy paste your graph, which I'm actually going to get rid of here, but you'll copy paste the graph that you created, and you'll also take the data table to insert into the template document. Your template document looks something like this. You'll see you have instructions, right? There's the data sheet in case you need it. Just remember to make a copy for yourself. You're going to create the data table and the graph, and then provide figure captions. If you're not familiar with figure captions, there's a resource right here. Please make sure you're reviewing it. So you can see, right, I gave you the full explanation of how to do table and figure captions. I'm not going to sit here and go through that with you right now, but you should read this over make sure you understand the formatting. All right, I'm telling you right now, if the formatting's wrong, when I go to grade these, yeah, you're gonna end up failing it because it's just very, very simple, and I'll show you how to do it in a moment actually. But it also walks you through what do you actually put into it, what's the thought process, the organization, the structure, if you will. And here again are some examples. Okay. But let's jump back over here to your template. You'll see this is the box where you're going to go ahead and enter your table. You'll place your graph in here. There is a rubric for you to review. So I'll fill that out with you. Green means go. If you get something like this, for example, that means that you did everything you needed to. You got the high marks for that particular category. So a green here would mean you got the highest possible marks of 10. If you had a yellow, that means slow down, you missed something. So you had most of this description, but not all of it, and you would have earned the 9. Okay, but let's put this back at transparent. And then here's what I'm expecting from you guys. So there's the table that I just copy and pasted from the example tab on the spreadsheet for light availability. And there's the graph we just made together. Note what I've added, though, is the caption, which is right here. So note that captions for tables always go above the table, where figures, like graphs, which is everything except for a table, the figure captions go below the graph in this case. Please make note of that. Note also that in terms of formatting, we're using size 10 font. It's bolded, and instead of doing what you normally do, like left aligned, I'm assuming, you guys want to switch this to justify, so it goes from end to end. That is the protocol that science likes to see, I guess. But you'll notice, right, we always start with table 1. If there was a second table, that second table would be table 2, and so on. But you do table 1 colon, and then this first sentence ends up being the title of your actual table. The next couple of sentences might be the description that kind of explains where that data is coming from and what you were doing, and then the last couple of sentences will be more of your analysis. Same thing kind of applies for figure captions, right? Figure 1 colon, and then this is the actual title. That we're not going to put that on the graph. The title always goes in the caption, but that acts as the title for your graph. The next couple of sentences are going to be the description, and then the last couple of sentences, really, like right about here, I think, would probably be the analysis, where you're trying to analyze what is this telling us. In this context, you want to talk about the error bars and what the overall message is of that data set. All right, so I hope that kind of makes some sense there. But yeah, that's kind of an overview of how to go about, doing your NPP student submissions. And I hope that little tutorial was helpful. Again, please make sure you're reviewing the figure captions, but I'll see you guys in class. Catch you later.